Hello, everyone. This is biology, and we are working on Module 7. Today, we're going to be working on the class notes for page 4 and page 5. Now, autosomes are chromosomes. Remember that human beings have 23 sets of chromosomes. It means there's 2 of each kind. One comes from the mother, one comes from the father. Now, 22 of those sets of chromosomes are autosomes. That means that they don't have anything to do with determining the sex of a baby. So these are the chromosomes that do not decide or determine. Decide is just easier to write. Sex of offspring. And that simply means whether it's a boy or whether it's a girl. Now, there's one set of chromosomes that are sex chromosomes, and these are one pair of chromosomes I guess I'm going to come, well, no, I'm going to do it on the first. I'm going to run out of room, so here we go. Of there we go. Chromosomes that determine sex or gender. Okay, and on this pair of chromosomes that are sex chromosomes, there are other genetic traits that are coded for on that chromosome. And those are the sex-linked genetic traits. So if you were to do a Punnett square for the sex of a baby, a female has a capital X from one chromosome and a capital X from another chromosome. Capital X from the father, capital X from the mother. A male gets a capital X from the mother, but the father gives a capital Y. So let's do a Punnett square and look at this a little bit. Okay, we do a Punnett square. This will be the mother. This will be the father. Now, that means here we have an XX, XX. Here we have X and then the Y. Here we have the X and the Y. So you see the chance of a female, see we have, let me get my colors out here. Yeah, there we go. So here we have, for female, we have 2 boxes. Remember, each box is worth 25%, so it's going to be a 50% chance that the offspring is going to be female. And see, here's 2 boxes that are males, and it's a 50% chance that the offspring is going to be male. Now, these sex-linked genetic traits often are decided by, they're only decided by whether you're male or female. And we're going to do an example of this. Here we have a heterozygous red-eyed female Drosophila, which is a fruit fly. It's crossed with a red-eyed male Drosophila. Will any of the offspring be white-eyed? Okay, that's the recessive. So let's, the red eye is dominant, so we'll make that a capital R. The white, which is recessive, is going to be the little r. Now, the thing you have to remember with this, it's a sex-linked trait. The Y gene does not carry either one of these genes. It's like only the X chromosome has it, not the Y. So there's only one of these genes that's going to come through. That's why it's sex linked. So let's look at this. Okay, so the mother, remember, she has an XX, and she's heterozygous, which means she has different letters on the different, since it's sex linked, there's going to be a capital R over here and a small r over there, because they're linked to the sex chromosomes. Okay, the male is also red-eyed. He's going to be XY, and remember, the Y gene cannot carry the R, so it's going to be a capital R. So let's do a Punnett square and see what it turns out to be. Okay, so the mother will put down here capital X with a big R and a capital X with a little r. And over here's the father, so we have a capital X with a big R, and we have a Y, which has nothing because it doesn't carry this gene. So now we cross them, we have X, R, R. We have X, big R, X, little r. Here we have X, big R, Y. And in the last box, we have an X, little r, Y. Okay, so let's look. Remember, if it even has one of these capital R's, it's going to be red. So this has a capital R, so this is going to be a red eye. This is a capital R and a little r, so you'll see the red. The capital R covers a little red. Okay, this is a male, but it's going to have red eyes because it has capital R. But this last one has just a little r. There's no capital R to cover it up. So this one is going to be white-eyed. Okay, remember each box in the Punnett squares were 25%, so there is a 25% of a white male. Now all the female offspring will be red-eyed, and half of the male offspring will be red-eyed. But out of all the offspring, 25% of them should be white males. Now we're going to talk a little bit about polygenic inheritance, and that's that genes aren't always quite as clear-cut as the dominant you see and the recessive you don't see. Sometimes the genes mix together or affect each other in unusual ways, and that's what polygenetic inheritance is. So what you see, your phenotype, what you see, is affected by the interaction of genes. It could be 2 genes, it could be more than 2 genes. So it's 2 plus genes, not just 1 gene, but several genes. An example is like skin color. And you can see this in real life. You know, sometimes people have dark skin, like the mother will have dark skin and the father will have light skin. And the babies will have sort of a mixture of color from each one. They'll have like a medium colored skin. So it's an interaction between several of the mother's genes and several of the father's genes. One just doesn't cover it up. Okay, so there's a couple different kinds of this. We have apostasis, we have pleiotrophy, we have incomplete dominance, and down here we have codominance. Okay, so those are the 4 that we're going to be talking about. Now, they're all types of polygenetic inheritance. Now, apostasis is simply that one set of alleles affect how another set is expressed. For example, in rabbits, there's a gene called the blue-eyed white gene, okay? And that means that they're going to have blue eyes and white fur. If this gene is present in a rabbit, it's totally dominant. Underneath, however, is a different gene for a different fur color. So this will cover up any other genes. Totally dominant. If you have a rabbit and it's blue-eyed white and you want to figure out what genes are underneath that blue-eyed white gene, you have to breed it to something else. If you want the blue-eyed white rabbits, you'll have to breed it to a blue-eyed white rabbit. Pretty interesting. Okay, another one is called incomplete dominance. So if it's heterozygous, if it's a heterozygous genotype, that means that the letters are different. They show a mixture. Between the two, between the dominant and the recessive. We'll say alleles. That's easy to fit into this space. Okay, so an example is a snapdragon, which is a flower. Okay, in a snapdragon, if you cross a white snapdragon with a red snapdragon, you're going to come out with a pink snapdragon. Okay, it's going to be a mix between the two different alleles. So if red is dominant and it's like this is actually what you're going to see as pink. And the next one is pleiotropy. And this is where a single gene can affect many different things, many different phenotypes. A single gene affects different phenotypes. Now, there's a couple typical examples of this. One of them is albinism. Albinism is a gene that causes a person not to have any melanin in their bodies. Now, melanin is what gives your skin color, gives your eyes color, so that not only will your skin be very pale, but your eyes will be red because there's no color in them. Usually your hair is white just because there's no melanin. And it affects many different traits in your body. Another one is sickle cell anemia. It affects the shape of your red blood cells, but it can affect other things in your body as well. Now, the last thing that we're going to talk about is codominance. Codominants are that the two alleles are different, but neither one is considered dominant. Now, the typical example of this is blood type. Okay, so you can have a blood type A, you can have a blood type B, and you can have a blood type O. Okay, A and B are codominant. Let's see, we'll put codominant. That means that neither one of them covers over the other. Oh, you can also have an AB blood type. So if you have an A blood type, you can have A. A is your genes, and you can also have AO. because O is the true recessive. So B blood type, you can have BB, and that would be homozygous, or you can have BO, and it's still considered to be a B blood type. Now, an O blood type is simply OO. This is the recessive of this gene. And then if you have an AB blood type, your genotype is simply going to be AB. Now, the reason this is important and why it's important to know your blood type is because if you're going to get a blood transfusion, you have to get your own blood, your own type of blood. Because on the outside of your blood cells, there are protein markers, and these protein markers are antigens, and they trigger the production of a substance called an antibody, which will attack your cell. Will attack the other cells as being strangers. It's supposed to be something that helps your body fight against infection and against strange cells, but in this particular case, it'll fight against the blood that's put into your system. So that's what an antigen is. It's a protein that triggers the making of an antibody, and the antibody will attack whatever has triggered it. Now, if you've ever heard of Rh factor, I'm sure you've heard of like A+ blood type, A- blood type. Well, the Rh factor is simply another antigen, another protein marker that exists on blood cells. Okay, if you have that marker, you're a plus. That means you have it. If it's a minus, it means you do not have that marker. It's another antigen that has to be taken into account when you're giving blood transfusions. Okay, we'll say another antigen on blood cells. So that's where the plus and minus. So you can have an O blood type and be O minus, and that means you completely have none of these markers. So now we're going to take a few minutes and we're going to look at genetic disorders and diseases and what causes this. Now remember, everyone is given 2 genes. For example, you can have a gene for, say this is for a disease, but what we're going to say is that the little d is the no disease. No, excuse me, that's backwards. The little d is the disease. Okay, that'll give you the disease. The big D is no disease. So it's recessive to get a disease, but if you have a genotype like this, you are a carrier for the disease. It means that you don't see it, but that you do carry it in your genes. So a genetic disease carrier is a person who has a recessive disease allele, but it's covered up. It's covered up, and the only way it can be covered up is if they're heterozygous. That means they have 2 different genes. Okay, an example of this is color blindness. There are some people who have the gene for color blindness, but it's recessive, so it's covered up. So, like, for example, their gene type would be this. The capital C means that they can see colors, but they're carrying this color blindness, and if they get married to someone who's colorblind, then many of their children will be colorblind. Another thing that can affect the diseases you inherit, and there's some diseases that are only found on the autosomes. And remember, the autosomes are chromosomes that don't have anything to do with sex. So this is just genes that are not on sex chromosomes. So you can have genes for disease that are not on the sex chromosomes. They won't be affected by whether an offspring is a boy or a girl. An example of this would be lactose intolerance. Doesn't have anything to do with whether you're a boy or girl. But then there are some genes that are connected to the sex chromosomes that cause disease. This is on genes that are on the sex chromosomes. And an example of this is hemophilia. Okay, hemophilia is a disease connected up to the sex chromosomes that causes your body not to be able to clot, or your blood can't clot. Let me write it better. So if you could get a cut, the cut will keep bleeding and bleeding and bleeding and bleeding and bleeding. It will never make a scab. Now they have medications that can help with this nowadays, but this is almost entirely afflicts males because it's connected up to the X chromosome. Or the X and Y chromosome. But, females can be carriers, but it afflicts males. Okay, mutation. A mutation is a radical change in an allele. And oftentimes we don't know why this happens. And when you have a radical change in those nitrogen bases that are in the middle of DNA, because that's what's happening here, those bases are getting changed, you come up with a disease. And because the proteins can't be put together correctly. An example of this is the full name is Hutchinson-Gilford progeria syndrome. I'm just going to put Hutchinson-Gilford disease. And this is a rapid aging disease. Where children their bodies, they don't get real big, but they do show signs of aging. Now, there's two different ways that you can have some of these happen. One is actually in the change of the chromosome structure itself. Another is a change in the chromosome number, and both of these happen during meiosis. Whoops, meiosis. Remember, meiosis is the dividing of the cell into gametes. And that's really when you have mutations or things happen, this is going to be the problem where it happens, meiosis. Mitosis, it might happen, but it's not going to affect your sex cells. In other words, you're not going to be passing it on to your offspring. Except if it happens in meiosis. So you have change in chromosome structure. This is a chromosome itself may lose a part of its structure, or it gains structure somewhere along the line. So a chromosome loses or gains genes. You're not losing the entire chromosome, you're just losing or gaining pieces of it. An example is that it's called cat's cry disease. And what happens is the fifth chromosome loses some genes. Cat's cry disease is one of the symptoms of it in a baby is that they cry like a cat instead of like a normal human baby. It's a very different kind of cry. There's other physical things that go on with cat's cry disease as well. So here you change the chromosome structure, but you can also lose or gain whole chromosomes. And that's what happens here. So you lose or gain chromosomes in meiosis. So normally you're supposed to have 23 pairs. You might come out with 22 pairs, or even instead of 46 chromosomes, you're missing one. So you could come out with 47 or no, 45 chromosomes. Something has happened. Instead of splitting evenly, they split unevenly. An example of this is Down syndrome. Where you have an extra chromosome. Okay, let's go on to page number 5, and we are going to talk about gene technology. Now, there's some interesting things that science is doing with genes now and with DNA. They can splice genes together, put them into different organisms, and a lot of this has been done especially with bacteria and plants and some animals. And they're doing it in order to improve life for us and improve the nutrition in foods, uh, increase disease resistance in different organisms. So that's what we're talking about right now. So gene technologies. Now, a genome is a map of the genetic information that's encoded in the DNA. So basically, it's a map of the DNA. What does it encode for? Now, in April of 2003, the Human Genome Project is a complete map of human DNA. They may not understand all of it. It's still very much under investigation. They don't understand what all those different base pairs do. But they at least know where things are decided. Like these genes code for eyes. These genes code for skin color. These genes code for, like stomach cells or whatever. These genes code for heart cells. They have a much better understanding of that now. Okay, they know that base pairs, remember that's C and G and A and T, adenine and thymine, guanine and cytosine. There are 6 billion of these in your DNA. Those 6 billion base pairs make up 60,000 genes. That means they're coding for 60,000 different proteins. And those 60,000 genes are broken up into your 23 pairs of chromosomes. Now remember, this is divided into coding DNA and non-coding DNA. And the coding DNA is what decides proteins. what they know besides proteins. They don't necessarily know what everything else is doing. They're still discovering that. Now, the coding DNA is 99% identical in every person, but it only accounts for 2 to 3% of DNA. It's still a lot of DNA, but it really only accounts for 2 to 3% of it. That means that there's 97 to 98%, which is non-coding DNA, and this is all different in different people. Now, the cool thing about this is that it matches up. This is passed on from father to child or mother to child, and so we can use it for identification, both in crime solving and in matching up families, because it's different in different people and different in different families. But there's some terms we need to understand before we can really talk about what they're doing, and like DNA profiling and genetic engineering. Okay, so first thing is you have short tandem repeats on the DNA. And this is short sequences of this non-coding DNA that get repeated different numbers of times in different people. So they're sequences of non-coding DNA and they get repeated in different numbers of times in different people. So, and these all different in length in different people. And we use this to do DNA profiling, which can be used in crime solving CSI. It can also be used in testing for family relations or identity. Now, also on DNA, okay, not on the DNA, but they use these restriction enzymes in order to cut these short tandem repeats. And restriction enzyme is unique to one sequence on the DNA. So we'll put that here. Before I define it, it's unique to one DNA sequence. Like it knows where the beginning and the end is to that one sequence, and what the enzyme will do is it can recognize and cut that sequence out of the DNA. So it recognizes and cuts one sequence out of DNA. Now it has what they call a sticky end, and it has a blunt end. Okay, so use number one, the sticky end is used in genetic engineering because you want to stick things together. It's a good way to remember it. So, engineering. And the blunt end is used in this DNA profiling. How many of these restriction enzymes have been discovered? Over 300 of them. Remember, and each enzyme only cuts out one sequence of DNA. So if you picture it, you have like a long chain of these letters, because DNA is a bunch of letters put together, and these restriction enzymes go through and they cut off pieces. So now you've got the DNA cut into pieces. So let's talk about what they do then. We're going to talk about DNA profiling. And this is used a lot in forensics, which is crime solving. Step number one is to cut the DNA with restrictions enzymes. Okay, step number 2 is called gel electrophoresis. 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 And what gel electrophoresis does is it sends all these different pieces into a gel-like medium, and this sorts it into size. And then in step number 3, these different sizes get compared. Now, the chance of someone who is unrelated to someone else having the same DNA profile is 0%. So, like someone you're related to, you might have like a 70% match. That would imply, or an 80% match, that a lot of these that you have inherited from this person or co-inherited if you're brother and sister, 70 to 80% of these non-coding DNA fragments are the same. That would imply that you're related to each other. Now, can you think of the one chance where you would have a higher, almost 100% match? Identical twins. Identical twins start with the same DNA profile, so they will have very close to 100% match. Now, sometimes like in DNA profiling, they only have a very small sample of DNA, not enough to really send through for gel electrophoresis. So what they do is they use this process called polymerase chain reaction. If you remember, polymerase is an enzyme that helps to create DNA. So what they do is they send it in, the polymerase chain reaction is a process that's used to copy DNA many times. So if it copies DNA many times, now you have a sample of DNA that you can use for DNA profiling. You have enough of it. Now, the last thing we're going to talk about is genetic engineering. And I don't think that we really even can comprehend how much in our world is affected by genetic engineering. So what it is, it's we're taking out pieces of genetic information from one organism, growing them, and then inserting them into the DNA of another organism for whatever reason. And it's made possible because these different genomes are being mapped, so we know which genes cause what thing to happen in different organisms. Because not only the human genome has been mapped, but many other organisms have had their genome mapped as well. Now, genetic engineering takes advantage of what's called a plasmid. Okay, so in a plasmid, a small section, it's a small section of extra DNA, which can be removed and reproduced outside of the bacteria. Now, recombinant DNA is simply the process of putting DNA into a different species. So you put DNA from one species into the DNA of another species. Now, some of the uses it's been used for throughout the years, it's made human insulin. Very important breakthrough in 1978. Lots of other medications and vaccines. If you've ever heard of microbes that break up oil in oil spills, they degrade the oil, that was developed by genetic engineering. Another thing that this has been used for is increasing plant nutrition. Making them plants more nutritious or more resistant to certain diseases or certain pests. Now, two more terms that you'll often hear as genetically modified organism. And this is simply refers to an organism with its DNA altered by humans. So the DNA has been changed by people. A transgenic organism simply carries DNA from another species. And you can look at my diagram. I think it's really good. You can also look at the explanation in the book if you want some more information. This just gives you an idea of different ways that we're using this knowledge that we've gained about genetics. And this is just, breaking stuff. This is a highly technological field. Lots of developments happening. If it interests you, look into studying this. There's a lot of people that go into crime solving and learn about this. But it also comes with a lot of controversy, especially genetic engineering. People question the morality of this. For example, if human beings become genetically modified, how is that going to affect a person's quality of life? Will they have diseases? If you're changing the genetics in plants, even though it seems like you're increasing the nutrition or increasing it in a good way, how do our bodies react to that? Do our bodies have more allergies because of it? Another controversy about the whole thing is if we change organisms and then put them back into a natural environment, will they become an invasive species? How will they affect the habitats and the organisms that are already in those habitats? So those are all controversies about genetic engineering. Thank you so much for listening. That's the end of our video for today. If you have any questions, please let me know.